when you estimate the intelligence of something, you're really just taking an IQ test yourself, because what you're saying is, this is what my intelligence and my imagination has allowed me to see in this system. And we should never think that, well, that's all that it has, right? There may be many other spaces within which it can exhibit intelligence, and we may simply have not tested it in the right way.
A unique guest requires a unique introduction, so I asked ChatGPT-4 to write one for me. Here goes. Welcome to the Smarter Not Harder podcast, where we explore groundbreaking ideas and revolutionary research with the world's most innovative thinkers. I'm your host, Dr. Ted Achacoso, and today we have a very special guest joining us, Dr. Michael Levin, an acclaimed American developmental and synthetic biologist at Tufts University. As a Vannevar Bush Distinguished Professor, Dr. Levin has made a remarkable impact in the field of biology, challenging the limits of our understanding and pushing the boundaries of scientific discovery. In this episode, we'll dive deep into Dr. Levin's research as he shares insights into three $64 million questions he is trying to answer. Number one, can we unlock the secrets of limb regeneration to revolutionize the future of medicine? Number two, how can we harness the power of cellular communication to control cancer growth? And number three, what are the ethical and practical implications of creating programmable living machines known as Xenobots? Dr. Levin's work on limb regeneration seeks to unravel the mysteries of regrowth and tissue repair in animals, with the potential to develop innovative therapies for human health. Imagine a world where lost limbs or damaged organs could be regenerated, transforming the lives of millions of people worldwide. Meanwhile, his groundbreaking research in cancer control investigates the language of cellular communication, looking for ways to manipulate cell behavior and halt the spread of cancerous cells. This pioneering approach could lead to new strategies for cancer treatment, vastly improving patient outcomes. Lastly, we'll explore the fascinating world of Xenobots, programmable living machines designed by Dr. Levin's team. The microscopic biological robots have the potential to revolutionize industries like medicine, environmental cleanup, and even space exploration. However, the ethical implications of creating and controlling life forms like these are complex and thought-provoking. Join us as we delve into these captivating topics, learning how Dr. Michael Levin's groundbreaking research is shaping the future of biology, medicine, and ethics. This is an episode you won't want to miss.
Hi, I'm Dr. Ted Achacoso, your host for this episode of the Smarter Not Harder podcast. My guest today is Dr. Michael Levin. Michael, hello and welcome to the show.
Hi, Ted. Good to see you. Thank you for having me.
Thank you for having to talk with me again. We spoke a while back, and this time around we're formally speaking in a podcast. Michael, Picasso famously said, "I hate computers. They can only give you answers." Well, it seems that it was Picasso who gave you the answers this time around. Can you tell us, what are Picasso frogs? What is the underlying big discovery underneath the Picasso frog experiments? And what is the significance of this discovery?
Yeah, the big central interest in my group is diverse intelligence. So we're interested in how different kinds of minds can exist in the physical world. And part of that is asking questions about how to identify intelligence and different kinds of cognitive capacities in unfamiliar places. So we look at all sorts of weird things that are not brainy animals, all the way up from molecular networks, all the way up to through tissues, organs, cells, all these different things, and look for ways in which we might be missing how much intelligence they have. So with respect to the Picasso frogs, the idea is that normally a tadpole becomes a frog by rearranging its face a little bit. So the jaws have to come out, the nostrils have to move. There's a number of movements that have to happen. So typically the way that people viewed this transformation is that as a kind of a hardwired response. In other words, every organ moves the correct in the correct direction, the correct amount, and that's how you go from a standard tadpole to a standard frog. And so that process doesn't have to be very intelligent. You don't need to know anything. You just move in the right way, and that's it. So we decided to test the hypothesis that it actually has considerably more competency than that. And one way you can test our definition of intelligence is one that William James used, which is the ability to get to the same goal by different means, and with different degrees of competency. So we simply ask the question, okay, is it just a rote set of movements, or do these subsystems actually know where they are and they have some idea of where they're going? And the way you test that is by interventions. And so what we did was we created these Picasso frogs. And so we created these tadpoles where all the organs are in the wrong place. So we introduced, so this was the work of Danny Adams in my group, and what she did was she basically introduced some RNA into these animals that caused the various components of the face to come out in the wrong position. So the jaws might be off to the side, the eye might be moved to the top of the head, everything is kind of scrambled. And the remarkable thing that we found is that those animals largely make pretty good frogs, because all of these things move around in novel paths through novel distances. Sometimes if they go too far, they actually come back a little bit, and they move around until they land in a proper frog configuration, and then they stop. So this is important for a number of reasons. The most obvious thing is simply that it's showing us that yet another, and there are many amazing examples, it's showing us yet another example of this situation where the body and this whole process of morphogenesis, so cells, cellular collectives being able to build and maintain complex forms, and in some cases repair them, that this process has some interesting competency to resist perturbation. So so this is another example of that. And like I said, there are many other examples. This is one example. I think the bigger lesson for us here, and we've had other studies that have reinforced that lesson, is that you can't simply assume the level of competency of anything just by sort of sitting back in an armchair and philosophizing about it. You have to do experiments. And until you check, you don't know how smart anything is and what competencies it has. And the way you find that out is by perturbing it in various ways and seeing what it does. So I think that's kind of the lesson we learned there.
Perfect. I just as an aside here, I really love the definition of artificial intelligence that you're using, because I know that a lot of your work is now actually within the artificial intelligence realm. I wrote a paper way back, and I said we should sharpen the definition of artificial intelligence. We should actually include artificial ethology, because if I see a baby in a room, I really don't know that the baby is intelligent, unless it does something intelligent. So it has an exhibition of behavior, in the same way that all of these Picasso parts for a frog, actually moves or behaves intelligently outwards. Just an aside where I remember that I actually pondered on the definition of intelligence. Instead of just artificial intelligence, are we having artificial ethology? But I just recalled it from the definition that you just gave here. Now,
I think that's absolutely.
Go ahead, please.
Sorry, I was just going to say, no, I think you're absolutely onto something very important, because for two reasons. One is that we are very primed, our cognitive system is very primed to recognize intelligence in traditional behavioral space, so the three-dimensional space around us. And so we're very used to looking at middle level, sort of medium scale objects moving at medium speeds and doing interesting things, and we recognize agency and behavior and so on. But imagine for a moment if you had like an innate sense of your blood chemistry, right? If you had a sense that was facing inwards and you could tell what your blood chemistry was, I think you would have no problem visualizing what physiological space was. You would have no problem realizing that your liver and various other organs are behaving, have various competencies to behave in that space, to navigate that space. And I think we have to be very humble when we the big thing I harp on is this idea that when you estimate the intelligence of something, you're really just taking an IQ test yourself, because what you're saying is, this is what my intelligence and my imagination has allowed me to see in this system. And we should never think that, well, that's all that it has, right? There may be many other spaces within which it can exhibit intelligence, and we may simply have not tested it in the right way. So I think it's important to stay humble about that.
Right. When you said that statement in a lecture, actually, where you making an IQ test on yourself, it's really our way of probing, what we know and we don't know on ourselves, rather than on what we're looking at. For me, that's very, very important. Is that the level by the level, or by which we are actually saying this particular layer is intelligent. So the agency, as you said, where is the agency coming from? Is it from the cell? Is it from from the organ? Is it from the organism itself? And that has been a very big component in my thinking about network systems, because that's what my work was in artificial neural networks. The second question I have for you is going with the frog model. I understand that you were able to grow anatomically correct and completely functioning eye in the belly of a frog, not by manipulating DNA, but by manipulating bioelectric circuits using ion channel proteins. What other organs have you created using this technique, and do you foresee this technique as transferable to growing human organs?
Yeah. So we have made so far. We have made forebrains. We have made hearts. We have made otocysts, which are basically like the inner ear. They're kind of a balance organ. We've made legs. We've made tails. I think that's it. There are many other things we haven't yet figured out how to make. I certainly think this will be applicable to human medicine. These kinds of mechanisms are extremely well conserved. Human patients with mutations in the channels that control these bioelectric signals have exactly the same kinds of mutations that you see, the same kind of malformations that you see in the animal models. So this is in no way a frog-specific thing. I fully expect this to be human relevant.
Michael, many years ago, shows you how old I am. We used to think in medicine or in surgery or in organ transplantation, at least the original thing was that we had to put the organ, like for example, the kidney or the pancreas, where its original blood supply is. And then the thinking evolved such that, no, you can just put it in any blood supply that is actually safe and accessible should anything happen to it. And so we began to transplant kidneys elsewhere, and we isolated beta cells of the pancreas, put them in sort of a bag, and made them function, in the wrist or somewhere that's accessible. And when you create this for humans, are you envisioning the same thing? Because it was a seeing eye, right, that you were able to create. That's all the connections and anatomically correct, but it was ectopically placed, but it had a full function. And so do you envision that, for example, someone, the liver is a highly generative organ, but some people, from cirrhosis of the liver would actually need a liver transplant. So do you envision it that it can be placed in another space, other than where it is now, so they don't have to dig out the old liver and put it in, put in grow a new liver?
Yeah, that's a great question. So we have seen in the bioelectric code, which is one of the things we work with, we have seen that in some cases what the code specifies is local, let's say, tissue or organ level pattern. And in other cases, it specifies very large scale anatomical decisions, such as build whatever normally goes here. So I'll give you a very simple example. The ion channel modulators that we use to grow a tail that's been lost or a leg that's been lost are exactly the same. Exactly the same.
Oh, I see.
So we are not in that particular case. We are not. There are other cases, but in that particular case, we are not telling the cells build a leg or build a tail. We're simply saying you already know what goes here. Go ahead and build it, and you never get a tail where the leg goes. You never get a leg where the tail goes. So there's a couple of different versions of our technology that I could imagine happening in the future. One is that you could imagine either locally somewhere in the body or even in a dish, you could grow the organ and then transplant it, as you say, into the wrist or some other accessible area. There's another version of this, which is applied to the correct location in the body. And that just says rebuild whatever used to be here, whatever belongs here, rebuild that. And in that case, you would want it back in the correct, in the correct location. I think both of these should be doable.
Okay, because I have some kidney transplant patients here, and there's always the fact that if the other kidney is not infected, or is not really doing any harm, it's simply not functioning, you could actually just put in another another kidney in there. So the person can have 3 kidneys and one
Yep.
is fully functional. So, we could do that. But I also seen kidneys that have been placed in other areas with a different vascular supply. So
Yeah.
this is actually encouraging. You have the choice, and
I think so.
I'm saying this tongue in cheek. It's like you could use certain organs in your body to enhance your aesthetic look, but we're not going to go there. Anyway,
Well, I mean, the enhancement idea is interesting because one thing we know, for example, is that brains are very good at reusing real estate, right? So if somebody loses vision, that area gets taken over by something else. So you can imagine, right, that if functionality is so good at taking over new real estate, you could imagine a solution where somebody gets a third hemisphere. Why not? We could do it in an amphibian model. We could do it tomorrow. You could graft in a third hemisphere. It would be completely functional, and over time, it would be really interesting to see if it would get used, and if so, what would it get used for? Right? Which of your many functions would take over this amazing new hardware that you now have? So I think augmentation is certainly on the table in the future.
Yeah, can we have our teenagers have rapidly developing forebrains which are already myelinated, so that you don't have to reach age 25 before the myelination occurs, so they can make better decisions. There's such a thing called I read this a while back, the organic teenage brain syndrome, where during the remyelination that occurs during the teenage years, and the hormones are raging. The hormones are actually causing a lot of cross communication among the unmyelinated fibers. And when I was looking at their work, and I said, well, we can create a forebrain that's actually more rapidly myelinating than this one's. So that we skip that very temperamental phase of teenagers.
It's an interesting idea. You wonder what effect that would have. It's possible that period of sort of metastability and plasticity is maybe necessary for future creativity. Who knows, right? What you would get from an adult that skipped. I mean, I have no clue, but it's possible that an adult that had skipped that time may not be the same as what we're expecting.
Yeah. Well, that was actually said, well, let's create another forebrain, a second one. Yeah. Even it's a thinner layer. That can be the auxiliary forebrain.
That's very interesting, right? You could have one that makes good solid decisions, and then another one that you can use for the wild and crazy sort of creativity that you want later on. That's an interesting
right. And if you took a look at the older experiments of Antonio Damasio, where people with damage the medial prefrontal cortex, who cannot assess risk properly, they will play the game knowing fully well what the rules are, or they could not seem to deduce the rules, where everyone actually knows the rules and they keep on losing. And in real life, they keep on investing money where, it's really a losing game. And I said, can we use this technology to at least, grow a different part of their medial prefrontal cortex and actually address this issue of damage to that particular layer?
Yeah, very interesting.
My next question is, continuing with the frog model, I have watched videos showing regeneration of an amputated frog limb that's anatomically correct and fully functional. Can you explain to us, how does it know when to stop growing the limb? And do you foresee this technique as transferable to regrowing limbs?
Yeah. How does it know to stop growing is really the billion-dollar question here. Not just for the therapeutic applications, but also for the really fundamental understanding of a lot of important things, including cognition and other things. How anything knows anything is a really fundamental question. The model that we use most of all is a kind of anatomical homeostasis model, where you have a circuit that's a little bit like the thermostat in your home that basically is able to do this loop. It's a very simple cybernetic loop where it measures something and it compares it to the memory of the ideal state that it has. And then if the error is more than a certain value, then it takes action to try and get itself to where it's going. Now, this is extremely familiar, of course, to biologists and doctors, because of things like maintaining temperature, blood pH, metabolic state. That's that's very obvious.
I'm a biology undergrad, so I'm lucky. I understand you.
Yeah. So right. So this is, this is pretty typical. But the amazing thing about some of these morphogenetic examples like regeneration of a frog or a salamander is that the cells will work very rapidly to rebuild. They proliferate and they migrate and they do all these things until they rebuild something, and then they stop. When do they stop? They stop when the correct structure is formed. So one way to look at this, a lens that you can put on this problem, is to say that perhaps this is an example of anatomical homeostasis. Instead of remembering the range of temperature or pH, a scalar, a single number, maybe what it remembers is the rough outline of what the correct organ is supposed to look like, not down to the single cell level. Because if you swap cells, it's perfectly happy. It's anatomical. It's the anatomical features. So then two interesting things come of making that hypothesis. First is that it now suggests a research program where, well, now you have to figure out, see, with a thermostat, you know where the set point is stored and you know how to change that set point. And so it suggests a research program where you try to find where the memory is. You have to find it, you have to decode it, and you have to learn to rewrite it. And that's what we've been doing, and I can tell you about that.
Sure.
The other amazing thing that comes of it is that it makes a really interesting statement about possible therapeutics. Because when you change the temperature setting on your thermostat, you don't need to rewire your thermostat. You don't need to change out your heating system. What you're doing, you're not dealing with a mechanical clock here. You're dealing with a very special cybernetic kind of system where there literally is a goal. I mean, it's humble as far as goals go, and as far as human goals, of course. But it's a tiny little goal. It's a tiny little goal that you can reset, and then the whole system will work towards the new goal. And you don't have to change out the hardware. So that tells you that, unlike the general assumption of modern molecular medicine, which is that you have to go all the way to the beginning of the hardware, genomic editing, CRISPR, right? You have to. That's the only way you're going to make changes, is down at the hardware level.
The rewiring, right? You have to rewire, right?
That's it. You have to physically rewire. And so anybody who's been paying attention to the information technology revolution understands now, since the 50s, that why do we not program our laptops with a soldering iron? I mean, it's funny. Why don't we do that? Right? Because we are not dealing with the kind of thing you have to necessarily rewire. We're dealing with a system that you can you can exploit the system's intelligence in terms of reprogrammability and memory and in some cases, in the case of the thermostat, the goal directedness. So what it tells you is that maybe in a system like this, if you wanted to do something new, something different, let's say repair, or let's say just a completely different shape in the case of synthetic robotics, you may not have to rewire the hardware at all. Maybe there's enough plasticity that you can simply edit the set point towards which it works, and then the cells will do what they do best, which is to build it. So we've been studying this, and I think we've made some progress. And so in the examples where we've studied, we can now literally see the way in the same sense in which neuroscientists do neural decoding, where they can scan the brain and see certain things, right? They can sometimes see certain kinds of memories and certain kinds of future behaviors and things like that. I mean, it's a very young science still, but we can, in certain cases, we can now see that set point, so we can see what the system visualizes as the correct pattern. And then we can rewrite that pattern. We can decode it and we can rewrite that pattern. And then when you rewrite that pattern, if the system is injured, it will go ahead and build a new structure. Now, this is we're just at the beginning of all this. We cannot do this broadly for any system that you want, unfortunately, because that once we are, this is the future of regenerative medicine is to work with the intelligence of your body. Why should we be trying to reverse what this incredible process of morphogenesis to figure out what gene do I edit to give you this feature? I mean, who knows, right? When that inverse problem is too hard, but luckily we don't need to solve that problem, because that's not how biology builds bodies either. And so that's what we've been doing is building tools and actually sort of conceptual frameworks to try to find these pattern memories and rewrite them in place.
Yeah, I think that you were touching on what you called in some of your lectures, the anatomic compiler. Is that what you're working on?
I mean, the anatomical compiler is not something strictly that we are working on. It's more of a vision of where I think the field is going. So when when you embark on this research, you have to ask yourself, or at least I tell my students, you have to you have to ask yourself, what does the end game look like? In other words, how do you know you've succeeded? Where are we where are we going? What's the final goal? And to me, the final goal of regenerative medicine is a device like the Anatomical compiler. I don't expect to see the full thing necessarily in my lifetime, but soon. I think this is where we're going. I think what the Anatomical compiler is, it's a design challenge. What it says to you is, you should be able to sit down in front of a computer, draw the plant, animal, organ, whatever, a robot, whatever that you want. And what it will do is tell you the set of stimuli that have to be given to individual cells to get them to build the thing. Now, once you have that, what you know that you have solved this problem completely, because then you have ultimate control over an anatomical form. And so then now that gives you something to shoot for. And now you can ask yourself, okay, why do we not have it? What are the barriers? What needs to happen for us to have something like this? It's a target for the future of the field. It's not just one simple. Oh, and the other thing I need to say is that this is really important. A lot of people see the Anatomical compiler as some kind of 3D printer that will literally construct what you're doing. Whatever you're looking for. This is absolutely not what we're talking about. A better way to think about it is it's a translator device. It's a communication device. It's something that helps you communicate to a radically different intelligence, which is the collective intelligence of cells. And what it allows you to do is to communicate your goal, meaning I want a heart that looks exactly like this, to those cells so that they can go ahead and build it. It's not a 3D printer. It's a communication device.
There are two things that I wanted to say here. Is that one of your explanations that sort of like made me understand the discovery and the work that you're doing, and sort of like, wow, this is the wow part that I remember very clearly, is that when you said that DNA simply specifies the hardware, right? The cellular hardware, that like the ion channels, right? And ion channels are the ones responsible for creating the physiologic software, which is the bioelectric circuit by the flow of ions in these channels, which then becomes like an erasable programmable ROM. That dates me, but it's how I look at it. And then you could do various all sorts of things in the bioelectric circuit without actually touching the DNA. And as you said very clearly, and I hope this is actually clear to many students, is that it's a reverse engineering problem that is unnecessary, right? Because there's a layer in there that you could actually program. The second thing that I wanted to say is actually, hurrah! I've been saying so many years that because the DNA sits at the center of the cell, we are actually trained to take a look at the foreground while ignoring the background, kind of like the illusion where we see the vase without seeing the two faces that actually make up the vase, right? The two faces that are facing each other and make up the vase. And I've always been saying that our society has become too DNA centric, and the funding has been way too much into this DNA centrism, that we have forgotten that there are actually other layers in there that the body uses where you don't need to touch that code, right? And surely, as you said, in your other lectures, surely evolution has come up with a problem-solving system, right? That is redundant, that's fault tolerant, that doesn't require rewiring every time something happens, right? And so those species with a bit better problem-solving capacities that are more resilient, in terms of various inputs, and they can still get into an adaptive energy minima. That would be the one that would be propagating and reproducing as a species. And when you said that people are we said like we're not using DNA, I said yes, finally someone's actually saying enough of DNA centrism for a while. We need to examine the other layers of this hierarchy. Now, my fourth question to you is, you have created alien biorobots, which you have called Xenobots, and I have seen the video of their fascinating method of reproduction. Can you please explain to us what Xenobots are and how they reproduce? And have you created other alien biorobots, I mean, from cells of organisms other than frogs, using this technology?
Yeah. So one of the really deep questions that you might ask is, about the degree of creativity or problem solving abilities of your various cells. So the interesting thing is that development is so incredibly reliable that it kind of lulls us into a false sense of limitations. I mean, acorns always make oak trees, frog eggs always make frogs, and so on. And you sort of get the idea by watching this robust development that that's all they know how to do, right? That's of course what else would they do? That's the genome. That's what they're going to do. And you get this idea of a mapping of a genome to an anatomy that says, that Xenopus laevis genome, that frog genome, encodes a frog in some way. This is the standard view that you get. So we did the following experiment, and this is work that we did in tight collaboration with Josh Bongard's lab at University of Vermont, and then the biologist in my group who did this was Doug Blackiston, and then Sam Kriegman was a computer science student in Josh's lab. So we asked the following question. What if we took some cells and we released them from their normal constraints? We gave them a chance to reboot their multicellularity. What would they do? If we took them out of the embryo, dissociate them, put them in a new environment without the other cells present, what would they do? Well, here are some options. They could die. They could crawl away from each other. They could do nothing. They could form a flat monolayer like cell culture. Here are some things they could do. In particular, notice that we are not engineering them via any kind of genetics. So their genome is completely wild type, no transgenes, no genomic editing, nothing like that. No weird nanomaterials. Literally, we are engineering them by subtraction. We are taking away the other cells that normally. Now, what we did was we started out with skin cells. So these are ectodermal cells. They're skin, and normally you look at these cells and you say, okay, well, what they know how to do is how to be this boring two-dimensional layer on the outside of the animal, sitting there quietly, keeping out the bacteria. But now the question is, okay, well, in the absence of the other cells that basically do a kind of behavior shaping that forces these cells to do that, what would they do on their own? And so what it turns out, what they do is they come back together into this this round little shape, which we call the Xenobot. Xenopus laevis is the name of the frog, but it's a biobot, so we call it a Xenobot. And this construct does a number of interesting things. First of all, the skin, the frog skin, has little hairs on its surface that sort of rotate to keep a flow of mucus going down the body, and this gets rid of pathogens and bacteria.
The cilia, right?
Cilia, exactly, right, exactly. So what it does is it uses the cilia to move around. It uses them as little oars to move against the water, and so the thing starts zipping around. It starts swimming around. So it starts moving around. And it has all kinds of interesting capabilities. First of all, it regenerates. So if you cut it almost in half, it'll zip back up to the normal shape. They have all kinds of interesting behaviors where they change. They'll be going somewhere, and then all of a sudden they'll turn around and go somewhere else. They can go in straight paths. They can circle around. They can interact with each other. Perhaps the most amazing thing so far is what you alluded to, which is kinematic self-replication, which is that if you provide them with a source of loose skin cells into the dish, they basically do von Neumann's dream. They assemble copies of themselves from loose material lying around. They will run around. They will push these cells into a little ball. And of course, because they're dealing with an agential material, much as we were when we created the bots, that pile becomes the next generation of bots. And when they mature, they go out and they do exactly the same thing. So you have multiple generations. Now, Josh's lab. So Josh's lab used AI to model how various sculpting events would affect the behavior of the bot. And then Doug Blackiston in my lab did all the hard work of actually implementing those sculpting kinds of interventions and show that, yes, we can actually predict what these things are going, some of the things that they're going to be doing. So it's a combination of really a lot of innate behaviors on the part of the cells, plus the AI, plus the manual microsurgery that Doug was doing. So, kind of one of the amazing things about them, of course, is that it turns out if you have these Xenobots and we've made it impossible for them to reproduce in the normal fashion. So they figured out in 48 hours, they found a completely new way of doing it. As far as we know, no other animal on Earth does that. And what they challenge us with is the following: for every other model system or natural animal on Earth, if you want to ask why does it look the way it looks, why does it have certain behaviors, the answer is always the same. Well, because for millions of years in the past, it's been selected to do this or that, right? Okay, There's never been any Xenobots. There's never been any selection
Right.
to be a good Xenobot, right?
It's the first time ever that we're encountering the organism, right?
First time ever. So now you have to start to ask yourself, where else can these anatomical goals, so the attractors in anatomical space and behavioral space and physiological space, in transcriptional space, where else do they come from? If it's not going to be evolution, where is it from? So we need to, and we have some thoughts about it, but the key thing is that
what are, just to interpret a little bit, what were you thinking in terms of the fundamental rules? Where it is coming from? Well, why does it have these types of behavior? I know that there's the physics layer that you have to contend with, and then the chemistry, and then how does it layer, or how does it network in your mind in terms of uncovering the first principles for something like this?
Yeah. So I'll give you some thoughts about it. It's going to sound a little bit strange because this is not how people typically think about this stuff. But
I love strange.
Yeah. All of this is bending in that direction. I think the right way to think about this comes from the history of mathematics, and it's been with us for several thousand years. Imagine that you are trying to evolve a triangle of a particular kind. So you evolve so you go through, there's a bunch of selection to get the first angle right, and there's a bunch more selection to get the second angle right. You don't need to evolve the third angle. It's a free lunch. Right. It's given to you for free. Now that's kind of amazing. Why can evolution save all that effort? So now you have to ask yourself, okay, the fact that angles of a triangle add up to 180 degrees, where does that fact live? Or here's another, I use this example all the time. There's a thing called a Galton board, which is basically, imagine a piece of wood vertical like this. You take a bunch of nails and you bang the nails into it vertically. Just boom, boom, boom. You just bang a bunch of nails. Then you take a bucket of marbles or ping pong balls and you dump it into the top of this thing. And you can buy these on Amazon. It's a cool toy. And they go boom, 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 boom. They go, when they land on the bottom, what do you get? You get this beautiful bell curve, always. Exactly the same. You get a Gaussian, right? A nice Gaussian. So you can ask yourself, right? So we who are used to thinking about this in the traditional way would ask two questions. You would say, first of all, where is that shape encoded? I mean, it's a very reliable, beautiful shape. Where is it encoded? It's very specific. Where is it encoded? You imagine tackling that as a biochemist. You could analyze the wood, you can analyze the nails. Maybe for really advanced, you'll analyze the position of the nails relative to each other. You're never going to find it. That's not where it's encoded, right? So our understanding of what it means to encode shape is not complete. And the next question you would ask is, well, how did you evolve that shape? And again, that's not the right way to think about this. You might have evolved the board to take advantage of the aspects of calculus that give you a nice Gaussian, but you didn't evolve the Gaussian. So this goes back. Now, I'm certainly not the first person to point this out. This goes back to Plato and Pythagoras, who thought about these things deeply and realized that some of these things, and they were in particular thinking about the truths of geometry and number theory and things like that, they have an independent life that's separate from the physical facts of the universe. The distribution of primes, the theorems of number theory, none of those things change if the initial laws of the universe were different. Those things are, where do they live? I don't know. But,
yes, but you and I think I'm safe to assume that you and I belong to the minority camp that mathematics is discovered and not invented.
No,
and I know that's a minority position, but I've always held that position because, Witten once remarked when he was looking at the string theory and the math of it, he said, this is mathematics that was just dumped onto us as a gift and we still don't know, what to do with it. And that statement actually had a huge impact on me in thinking about mathematics and the fundamentals of geometry. Where are these things coming from? I was deeply fascinated by Darcy Wentworth Thompson's on growth and form, when looking at all this stuff, like, where is this coming from? And then most recently, it's interesting, you're familiar with the work of Nima Arkani Hamed, right? And he's basically saying, space-time is doomed, and he comes up with the amplitudohedron and, cosmological epitopes and stuff like that. And these are beyond space-time. I said, okay, is this where these geometries are coming from? Is this where the mathematics are coming from? And I think what he has done is to push us into thinking. It's like, what else can we discover in those cosmological epitopes beyond space time? And
Yeah.
when I'm listening to you now and hearing about your work or reading about your work, it just really reminds me. It's like we still, as you said, we still don't know what the first principles are, but you guys are already thinking about it, right?
Yeah.
Do you think this will be resolved quickly, or this will require a great deal of time and new math being discovered before we actually are able to have something even just pragmatic that we could use, to help us and other organisms here in this planet?
I think the pragmatic stuff will be discovered in a short time frame, because it's so easy to there are so many free gifts like this. So not just geometry and mathematics, but in computer science, right? Once you got an ion channel, and this ion channel, let's say, is a voltage gated ion channel, so it's a voltage gated current conductance. It's a transistor. Once you evolved an ion channel, once you have a transistor, you can make a logic gate. Once you have an AND gate, you can make anything you want. So when you have a truth table to one of these functions, did you have to evolve the truth table? No, all you evolved is the
No.
ion channel. The truth table you get for free, right? And
Yeah.
all the theorems of computation, you get all that for free. So there are so many of these things, and biology is so good at exploiting them that I think the practical applications are not far behind. What I think might take longer is catching up conceptually to this, to be able to have better, more mature answers to questions like, where is it encoded? Where does it come from? But I don't see it. It's not physical. These things that are really tripping people up nowadays. To get past that way of thinking about it, I think is going to take a while. And to really get serious about the idea that we can explore this Platonic space in the exact same way we explore other spaces. It isn't, some mystical thing that we can't understand. It has a structure. Mathematicians know this. I've seen maps of mathematics where, well, over here lives, the topology, and right next to it lives something. Okay, so they already know you can do things like this. So I think we need to really understand that what biology. Here's what I think. This is a controversial point, but it's what I think. I don't think that evolution is really searching the space, the very difficult and rugged space of micro states, all the little mutations. I don't think that's what evolution is searching. What I think evolution is searching is the much nicer, much more smooth and powerful space of how to use these various affordances that physics, computation, and mathematics give us. I think what evolution does is finds machines that exploit this stuff. So and that's a much nicer space to search, and I think that's why evolution is so incredibly robust. So learning to think that way, I think, is going to take longer. Although, heck, if space-time is doomed and people are adjusted to that, Don Hoffman's stuff and all that, if people can adjust to that, then they can adjust to this. So maybe none of this will actually take that long.
Yeah, as you can see, I've spoken with Don Hoffman, I've spoken with Carl Friston. And how do you think Carl Friston's free energy principle figures into this, first principles that we're looking at?
Yeah, well, first of all, to be super clear, I do not have the math or physics that Carl does, so I can't follow him to all the details. But he and I write papers together, and we talk a lot about this stuff. From my understanding, which is not as deep as his, I think that what he's got is extremely valuable because. Not only is it, the part that we use directly is this idea of surprise minimization and how cells and even networks have expected states, which means they minimize surprise and all of that. That stuff we can use. The deeper agenda that he and people like my collaborator Chris Fields are doing is something very interesting, which is that I strongly believe that we have to take the continuity thesis very seriously. Both evolution and developmental biology are extremely gradual, and it doesn't make any sense to say that you were, from embryonic development, you were a blob of chemicals, and then boom, there's a magic lightning flash at some particular point, and now you're a cognitive being. I think this is ridiculous, and I think you just have to realize that it's a slow and gradual set of changes that takes you from being able to operate in some sort of chemical space and metabolic space, and then transcriptional space, and then anatomical morphous space, and eventually three-dimensional space, and eventually linguistic space, and then who knows what's after that. But I take this continuum very seriously, and what I take Carl to be doing, among many other things, is providing a framework by which you can pull that way of thinking about it all the way down. So how far, right? So I've been working on this framework called TAME, TAME, technological approach to mind everywhere, and the centerpiece of it is this continuum of persuadability, where you have all kinds of diverse systems, and then you can ask some engineering questions about what kind of cognition they have. And so people always ask me, how far down does it go? Right? Is there a zero? Is there anything in this world that has literally zero cognition? And I think what Carl and Chris and maybe other people are doing is showing a rigorous mathematical framework where you don't paint cognition onto a perfectly good physics as a kind of like afterthought because you didn't know what to do with it. That's traditional panpsychism, and everybody critiques it because, look, the physics work perfectly well, but you're painting some kind of a glow, a cognitive glow onto it that it doesn't need. That I agree, that doesn't work. What they're doing is something different. They're trying to recast basic physics from the perspective of the information processing being primary. And that, I think, is that's the winning position. I think that's what's going to make this all make sense. Why the continuity actually works.
Well, just for the sake of our audience, what he just said there really is that a thermostat is a thermostat, and you don't need to bestow cognition on it for it to work. Is that a one way of looking at it?
I would say it differently. I think a thermostat is an example of basal cognition. I think thermostats have little tiny nano goals, and I think that if you don't recognize a thermostat, and we miss this all the time, we don't recognize these systems. We're not good at recognizing these systems. If you don't recognize its cognitive capacity, you cannot relate to it in the most efficient, most useful way possible. Right? If you've missed the fact that, look, we recently showed that gene regulatory networks can have 6 different kinds of memory, including associative conditioning. This has massive implications for biomedicine, use of pharmaceuticals, and so on. But if you just sort of sit back with your as people do with their commitments, philosophical commitments, to say there is no way a gene regulatory network can have cognition. They just can't. They just can't. That's it. Once you've said that, you've cut out a whole bunch of empirical progress that you could have had if you were to say, well, let's find out. Let's try training them in various ways. Let's try doing various things and actually see what the level of cognition is. So I think things like thermostats are absolutely on the spectrum. Now, then people ask the question of, well, how much lower can you go? What's below that? And I think that through their work on very basic physics, the paper on a particular physics that he has and all that kind of stuff, I think you start to see that cashing out things like quantum interactions, like basic particle interactions, there is already a really fruitful way to look at those things from the perspective of behavioral science. To me, this is what it's about. Somebody was saying to me recently that, well, that's just a metaphor. It isn't really true. I don't know what really true means. I think all we have is metaphors. Some are better than others. And by testing our metaphors, we end up with hopefully better ones. But so now the question is this: the cognitive metaphor, how much good does that do you? Does that stop at some point, or does that keep paying off dividends all the way down? And I think what Carl is showing us is that there are portions of the sort of bag of tricks that the behavioral science gives us, including things like active inference and things like that. They go all the way down. They're useful at the particle level, just as they're useful for humans and who knows what else. So that's what I think he's doing, and I think that's extremely valuable work.
You said something about, okay, what's lower, and I know you don't like to comment on anything because you see things as a spectrum or continuum, and you don't like binary categorizations. So what did you think of? You're familiar with old Stephen Jay Gould and Niles Eldredge punctuated equilibrium. So do you think, what do you think of that particular model? And is that because we couldn't see the intervening gradual changes leading to the punctuated equilibrium? That's why we actually, think that it's a punctuated equilibrium model of evolution.
Yeah. I mean, as with any kind of with any kind of a change. I mean, look, I'm certainly not saying that a human level cognition is exactly the same as a flatworm or anything else. Of course, there are different capacities, but what is chunked into these distinct categories are our paradigms for understanding them. It's not the thing itself is perfectly gradual. It's the tools that we have to deal with these things. So if you step back on a macroscopic level and you look at anything, the evolutionary record, the developmental record, you say, oh, look, at this point it didn't have this, and now it has this. A very sharp transition. Okay, but if you actually zoom in, of course, what you don't have is, a set of parents, a set of hominid parents that didn't have, responsibility or free will or whatever you want to say is human. And now their children, boom, now their children have it. That doesn't happen. It doesn't happen in development. It doesn't happen in evolution. There's always a slow transition. And what you have to do is decide what scale you're going to work on and what are the relevant tools that you're going to have for this, right? And people often talk about the paradox of the heap, right? So you got this pile of sand, let's say, and you take off one grain, and you say, well, is it still a pile? Yes. You take off another, and you say, well, then that's the same problem, right?
It's a self-organized criticality, right? It's called self-organized criticality in a complex system. So it's the last grain of sand phenomenon. Which grain of sand actually caused the avalanche?
And, there's that, but they go even further and to say, look, you have to assume that that there have to be sharp transitions, because otherwise you run into this paradox, right? And my claim is simply this: the heap is continuous. What's not continuous is the tools that we have to work with it. So if you call me on the phone and you say, I've got a heap in my living room, what I really need to know is, am I bringing a spoon, a shovel, a bulldozer? What am I bringing? And my tools, right, functionally. So I'm an engineer. So to me, all of these claims are protocol claims. They're all claims about manipulating and what is the right tool to manipulate. So you can tell me that it's a heap that's appropriate for a shovel, or it's appropriate for a bulldozer, or it's appropriate for something else. And those things are discrete, because that's how we know how to build things. But the heap itself could be perfectly continuous. And I think in behavior science, we have exactly the same thing. We've chosen to call certain things. I'm going to use the paradigm of associative learning for this, or I'm going to use habituation, or I'm going to see this as a form of long-term planning. And that's great. And those tools that we have, those lenses that we have for looking at these things are discrete. But but that's on our end. That's on the observer end. The phenomenon could be perfectly continuous.
Right. I have two questions, and I know that you're reticent to talk about the other. But first is, can you explain to us what embodied cognition is? And the second one is, what are your thoughts about consciousness and its emergence? And I know this is a part where you don't like to talk much about it, but how would you lay out a scaffolding into how to think about it?
Yeah. I can say a few things on this. Embodied cognition, I think, is the broad, to me anyway, it's the broader mission of understanding mind in its full diversity of embodiments. So as I was saying earlier, right, we're pretty comfortable recognizing intelligent minds and intelligent behavior in space. We recognize agency, and but there are all these other spaces, different types of spaces, where various weird systems like transcriptional networks, like physiological circuits, like collectives of cells, like groups of animals that function in the collective functions in some sort of social space. So it's the understanding of how intelligence can function in diverse spaces with diverse embodiments, meaning that it doesn't have to be a brain. The scale could be tremendous. Could we think about the evolutionary process itself as a kind of agent that tests hypotheses by generating different genomes and seeing how they do, right? So that's an enormous agent. We're not used to looking at that scale. We're used to looking at scales of things like us, or molecular networks, or photons doing a least action kind of thing where they choose the shortest path. The embodied cognition is this idea that you cannot assume ahead of time, just because you're unfamiliar with the system, what its cognitive capacities are going to be, and that we have to have general tools for detecting, because sometimes we don't even know they're there, understanding, relating to these very unconventional spaces. And that's long before we encounter aliens or alien life or any of that. Even just with what we have here, we have plenty to work with that doesn't have the same origin story as us, so it doesn't have to be evolved. With the many things to think about. So that's the first thing, that the embodied cognition, and then the consciousness. So I'm not somebody who says that there is no hard problem of consciousness. I think there is. I think it's a real. I think it's real. I think it's a real issue. The reason I don't talk about it much is that I don't have terribly many things to say about it that smarter people have not already said on this topic, and also I don't have a solution for it, but I can say a couple of interesting things. One interesting thing, I think, is that there are a number of theories now, theories of consciousness that are supposed to explain what is it about the brain that makes it conscious. That's the goal of these theories of consciousness. And a student of mine named Avery, we just made a list of them, and there's probably half a dozen well-known theories of consciousness that say this is why the brain is conscious. One interesting thing to note is that, as far as I can tell, and we haven't published this yet, but we're working on this now. As far as I can tell, all of these theories, if you believe any one of these theories, for the exact same reason that theory says the brain should be conscious, you should be ascribing consciousness to other organs in your body, because all of the basic things that these theories lean on are happening everywhere else. So people will say, neurons. Okay, what's a neuron? Bacteria in every cell in your body is doing many of the same things that neurons are doing. A neural network, great. Most of the cells in your body have an electrical network that very slowly are doing many of the same things. So I have not found anything in any of those theories that will tell you why the consciousness in the brain is not zero, but the consciousness in your liver is zero. I've seen no scientific way to make that distinction. So that's the first thing. I think to the extent that we believe brains are associated with consciousness, we should be perfectly open to consciousness elsewhere in the body. Now, people will immediately say, but I'm a unified consciousness. I don't feel like the seven other consciousnesses in here. Of course you don't. You're the one that speaks. You're the one with verbal communication. That's the one I'm talking to, right? If I knew how to ask your liver, and we're working on that, by the way, if I knew how to ask your liver, I might get a completely different answer. You're not aware of it any more than you're directly aware of my consciousness. We have to talk. Right. For you to have some evidence, the problem of other minds, you have to have some evidence that there's consciousness here too. You don't sense it any more than you sense the ones you co-inhabit in the body. So I think that's another thing I can say about it. Another thing I can say is that one of the reasons there is a hard problem is that never mind what a correct theory of consciousness is. Let's think about what's the format in which it gives answers. What does a theory of consciousness output? So a theory of gravitation or a theory of evolution or all these other theories, we know what they output. They output quantitative predictions in a particular problem space that all of us can observe objectively and compare to the results of our experiments. That's what they output. What would a theory of consciousness output? It's completely unclear, because if it outputs publicly observable facts, then it's a theory of behavior. It's not a theory of consciousness. So I think the closest we're going to get to this is that, unlike these other theories, all of that's what makes this consciousness problem special. All of these other theories output things that you can objectively test in third person. What a proper theory of consciousness would output is not a set of facts, but a protocol for making me feel the same, whatever it is that the theory says is this conscious state. So basically art. Basically, it's the product of art. So the goal of a poem or a piece of art or something else is to enable, it's a medium that enables me to feel some conscious state that the creator wanted me to feel for whatever reason, right? And so it's a protocol. It says, here's some inputs, and if you allow yourself to have these inputs, you should enter this particular state, and that's what the theory of consciousness should predict. If the theory of consciousness says I predict fear, then the answer should be not avoidance behavior and things like that, but it should be, here, do these three things, and then you'll feel, and you will experience first person what the prediction is, right? So it produces protocols. It doesn't produce facts. or numbers or anything like that. And I can see some other stuff, but that's that's a starting point.
One of the things that I did 3 decades ago was, when I was working on the neural networks of C. elegans, really my goal there is remove all the subnetworks that was responsible for feeding, for mating, for locomotion. And I wanted to see whether or not there was actual network that actually said, now I feed, now I mate, now I move. And I found no such network. So at that time, I was convinced that consciousness was an emergent property of complex systems. And then recently, I thought about it a little bit more, and I think I mistook consciousness for the sense of agency. And I think a sense of agency is different from consciousness itself. And when I've been hearing some of your lectures, it's more like, basically we could take a look at different levels and what particular sense of agency like a cell has, or an organ has. And then we as an entire organism would have a sense of agency. And so I'm back to square one. That's why when I spoke to Don Hoffman, I said, you convinced me that this is not an emergent property of complex systems, but rather, consciousness must be something else. I still don't know what it is. It may be an inherent property of systems. But like you, it's sort of like looking at the writing of Mark Solms, for example, where it's very geared towards the human species. I was looking for and we can diagnose that medically. When he says awake, alert, oriented, essentially summarizes a lot of his theory of what consciousness would be in a medical context. So I actually differentiate medical consciousness from the consciousness that we're talking about, which I call philosophical consciousness. And then there's part of me that's looking at what we call biological consciousness. So we're looking at this and we're throwing around the term, and I said, well, it has to be in a particular space or context that we are talking about consciousness. So my next question for you, Michael, is that you and I seem to share a fascination for worms. Me for C. elegans and you for Planaria. I wrote a book in 1992 cataloging for the first time the complete connectome of C. elegans, both of synapses and gap junctions, based on the electron micrographs of Nobel laureate Sidney Brenner. The preponderance of synaptic connections on the right side of the body made me wonder whether or not chirality was as deep as the number of synapses, since 80% of the worms that I counted on the microscope corkscrewed to the right. I read about your interest in left-right asymmetry, so I thought I would throw that in. You, on the other hand, are working on the Planarian flatworms made immortal by their capacity for regeneration. I understand that by manipulating their bioelectric circuits using ion channel proteins, you were able to create two-headed Planarian worms, one on each end of the body. You also have shown that without manipulating DNA at all, this two-headedness persists permanently, meaning if you chop off both heads, the body will regenerate the two heads, one on each end of the body, instead of the expected head and a tail in the wild-type Planaria. This is surprising. Now, what are the implications of this? Because these implications, these are huge. And I want you to explain to our audience, what are the implications of this experiment?
Yeah. If you so we developed a dye-based method for looking at the bioelectric states, meaning the resting voltage potential of cells in the body. When you look at a normal Planarian, you see a particular bioelectrical state that's associated with, in fact, drives one-headedness. So if you chop a Planarian into pieces, every piece has to answer the question, how many heads am I supposed to have? I mean, it's really not obvious at all. And by the way, which end does it go on, and things like that. And so there's a bioelectrical circuit that stores the memory of how many heads you're supposed to have. What we figured out how to do is to rewrite the information in that circuit. And you don't, because it's an active memory circuit, you don't need to change DNA for this any more than you and I change DNA when we learn something new. You simply provide inputs that alter the stable electrical state that is kept by this circuit. I mean, it's literally a memory. And when you do that, you can change it to, say, two heads. And then it's a kind of latent memory because it doesn't do anything until you injure the animal. When you injure the animal, the cells consult that memory and it guides them to build two heads, one on each side. So so I think there's a few implications of this. The first implication is that it's a direct proof of this idea that shape isn't just emergent. It's not just that there are rules and every cell follows the rules and out pops something complex. I mean, that happens, of course, too in other systems. But this is the evidence that, much like with a thermostat, there literally is a set point. How do we know? Because we can find it, we can decode it, and we can rewrite it. That isn't possible with systems that don't have a set point. So that's the first thing. So it helps to understand this whole system as a kind of primitive cognitive agent. The second, and including the fact that this memory is a counterfactual. People say, "Oh, brains are amazing because they let you have a memory of the future or the past. You can remember things that are not true right now, right? You can have a memory of things that are not happening right now." Well, guess what? The system is exactly that. It holds us a definition of what the worm will do later if it gets injured. It isn't true right now. Right now, you've got one head, but the pattern says make two if you get injured. So that dissociation between the future and the current state is usually what people think about in brains. They don't think about this in other systems. The third thing about this is that if you cut these two-headed animals, they continue to develop they'll regenerate in a two-headed form. So this is very interesting because they have a completely wild-type genome. You can imagine I throw them into the Charles River. The way they reproduce is they tear themselves in half and regenerate. So 100 years from now, some scientists come along, they scoop up some samples. They say, oh, look, some two-headed worms and some one-headed worms. Awesome, a speciation event. Let's see which genes got the change here. Speciation event. No genes got changed there. So this, right? So this tells you that if you ask the question, what determines the number of heads in a Planarian, it's quite subtle. The answer is not DNA, because clearly the same DNA is compatible with two morphotypes that are permanent. What the DNA does do is generate a physiological machine that by default, like a calculator, when you first buy it from the store, what does it say? It says 0, reliably. It always says 0. But it doesn't have to say 0. You can change it with future inputs. So what the DNA does is it builds a machine that very reliably goes to the one-headed electrical pattern memory, right? Just like animals are born with built-in instincts for behavior in three-dimensional space. Birds make nests and all this. So they have a built-in physiological mode, but it's an awesome circuit that you can actually reprogram. And I'm sure we can find all kinds of other things. We've made two-headed worms. We've made worms with heads of other species of worms. We've made planaria that don't look anything like planaria with the same genome. I'm sure there's many other things you can do, but it provides a really interesting perspective on this distinction between hardware and software. And it tells you what the role of the genome actually is. The genome doesn't encode the number of heads. The genome, and so we have to all of the terms that we're used to using, encoding, storage, genotype, phenotype. All these things have to be rethought a little bit because there's so much action in this physiological space. It is you can't think about it the way that we've been thinking about, let's say, protein structure, and maybe not even there. I'm not sure, but Andreas Wagner is amazing. He has some amazing work on this whole issue of arrival of the fittest, he calls it, which I think is very clever. And he talks about all these same problems for protein space. I mean, so we do anatomy, but you can see this even in protein.
While you're saying that, this has huge implications on how we think about evolution. Normally, we think there's a change in genetic structure, they mutate, they produce other species that are adapted to a particular environment. And, I begin to wonder then that how much of, what we classified as speciation is not really a DNA event, or even not a DNA event on the starting point.
Right. Afterwards, you see, right. So afterwards, this is really a kind of Baldwin effect, right? Assimilation, because afterwards, if the two-headed worms were to do well, and I mean they don't, but if they were to do very well, you could imagine that at some point you'd get an ion channel mutation that takes you to that two-headed state right away, and you wouldn't need the thing that we did, and so this would be, it would become assimilated into the genome, and that's a standard kind of assimilation. And people think about this for behavior. This is exactly the same thing, but this is behavior in anatomical morphous space. It's not three-dimensional behavior. It's basically the Baldwin effect in anatomy. And I think these things are all over the place. I think evolution uses this extensively. It's just there for us to find.
Yeah, this is a comic relief here, Michael. You could program the calculator to not 0 but 42, the answer to every question in the universe, right? My next question for you is, in one of your lectures, you placed bioelectricity as informational medium for anatomical homeostatic agents in the amorphous space, and your biological endgame is the creation of an anatomical compiler because genetic information is not enough. I pioneered a clinical specialty called Health Optimization Medicine and Practice, or Home Hope, which detects and corrects imbalances at the levels of the metabolome. So we're dealing with the metabolic space. Our clinical endgame is to shift from geriatrization to metabolic neotenization, to bring metabolite values of older people into the range of when people were 21 to 30 years old. Do you think that it would be possible to do this using bioelectric circuit manipulation, using electroceutical drugs?
It's a good question. I don't have any data on that at the moment, but I do think that I don't know whether the bioelectric system controls metabolism. I really don't know, but it's certainly possible. And I do think that other approaches that we've been talking about here, which is really taking advantage of the software side of things, of the learning capacity, of the decision making, these kinds of things. I think those things should absolutely carry over, and I think I'd love to see that explored in the metabolic space. So we've explored it in transcriptional space and in anatomical space, and of course everybody's done it in behavioral space. But metabolic space, I haven't really seen that yet. And I think all of these kind of techniques, which is basically it's basically just stealing all the tools of behavior science and using them in a different space. So training, and the simplest version of it is some kind of preconditioning, which I'm sure people have done. But taking all of these things from behavioral science and testing them out on these metabolic circuits, I would bet any amount of money you're going to see the same thing that we've been seeing, which is that, you can train these things and they have anticipation and they have habituation and they have the sensitization and they can, all these different kinds and see how far they can go, right? Do they have surprise minimization? Do they have, who knows, right? So that stuff, I would bet any amount of money that stuff would work. I don't know if it'll be bioelectricity specifically. The thing about bioelectricity, I mean, I know no one loves bioelectricity more than me, but bioelectricity is by itself not magical. The reason bioelectricity is important is because it is one way that cognitive dynamics work out at certain scales in biology, but it's not the only way. There are other materials that can do, other modalities, I should say, that can do it too.
The reason why I'm interested in this is actually severalfold, but there are two. The first one is that for my patients or for my clients, what I do is that you detect the subtle toxicities, borderline deficiencies in the metabolites, and then you actually balance them. You give the actual metabolite, or the source of metabolite back, and should be, and the question for me always is that, am I going to do this in perpetuity, right? Usually the answer is yes, because if you're not eating properly and so on, those metabolite levels will decline again. Or if you're continually taking in the toxin, then the toxin will always be rising. But the second thing is actually more. I'm interested as a neuroscientist, in fact, and as an interventional neuroradiologist trained in this area. One of the things that I was looking at in the neural networks was, these are all like, GABA receptors, et cetera, et cetera. I was interested in that in terms of schizophrenia, and, Carl Friesen and I were talking about this and said that, GABA is one of the key molecules, in fact, probably the key molecule that we should investigate in schizophrenia. Now, I've been, as a pharmacologist, I've also been experimenting with the GABA A receptor, and how can we do this to change the the brain to increase the inhibition, for example, in terms of too much connectivity, or too noisy in the brain, or too fragmented in its processing. How can we use a neurotransmitter like that in order to work on the chloride channel, right? It opens the chloride channel pore, it hyperpolarizes, and then it prevents the system from firing a little bit too much. So this is where I was looking at. Well, yeah, I think in one of your lectures you mentioned the use of epileptic drugs, right? And many epileptic drugs act on the GABA receptors, especially the GABA A. Spasticity is usually the GABA B receptor. So as I said, well, should we be continually using these anti-epileptic drugs, or is there a way of being able to provide this using an electroceutical drug instead, right? That actually works on the bioelectric circuit, so we don't have to keep giving these seizure medications.
Yeah. Every case is going to be different, but I will say that what we notice in many of our examples is that, much like in behavior, there are triggers that then change the decision making of the system, and then there's all sorts of autonomous things that happen after that. So, for example, our treatment where you trigger the leg regeneration in a frog, the treatment itself is 24 hours. The leg grows for 18 months. We don't touch it during those 18 months. It's enough in the first 24 hours. If I tell the cells, build a leg, and if they hear me, and if they understand what we're saying, and it shifts from scarring to building a leg, all the downstream cascades, all the transcriptional cascades, the epigenetic cascades, all that stuff is going to happen. We don't have to be there with that same, and that's very much in keeping with a kind of behavioral approach. Once you've told somebody something, you don't need to stand there and repeat it a million times. You might have to refresh it at some point, but you don't need to be constantly doing it. You push the system into a behavior, and it already knows how to do, right? It already made that leg once. It already knows how to do that. Right. So I think there's going to be tons of stuff like this where you provide a stimulus, you leave it alone for a while, and then maybe you have to come back and do it again at some point.
Right. Well, the thing that's sounds like too much work for me. We like to do things the easier way, is that, for example, you have to create something that has to bind to the benzodiazepine side of the GABA receptor. You have to bind to the endogenous ligand. You have to create all of this stuff. It's like, is there any way to just put this in a particular pattern that it produces a proper circuit, and you don't have to really bother about what particular subtypes the receptor has in order to open up the chloride channel and induce the effect? Next question for you is this: education in the clinical specialty of health optimization medicine and practice, which I pioneered, has a module on evolutionary medicine where we teach that in order to shift away from pathogenesis, or causes of disease, and into salutogenesis, or causes of health, we must shift away from organology and into holobiontology. I've heard you say that we are a collection of cells. For our audience here, a holobiont is exactly that. And at Home Hope, we consider the body as a network system of different cell species that are all cooperating and competing at the same time. We consider the gut microbiota, for example, which is 2.2 kilos in weight, as an organ in itself and is part of the holobiont. As I understand it, a holobiont is different from a superorganism, and in that a superorganism is made up of the same cell type, like in some slime molds. I bring this up because in evolutionary medicine module, we teach that cancer cells happen when they break off from the control of the holobiont network. As independent cells, they then proceed with their goals of survival and reproduction. Thanks to your experiments, we now know that the network that asserts control is really the bioelectric circuit. Can you explain to us how the bioelectric circuit can prevent a cancer cell from proliferating and metastasizing?
Yeah. One way I like to think about this problem is not to ask why we get cancer, but to ask, why is there anything other than cancer ever? Meaning that we were all individual cells at one point, some sort of amoeba-like thing, and they were very competent in handling metabolic goals, physiological goals at the level of a single cell. what do these single cells want to do? Well, each one wants to become two cells, and they want to go where life is good and eat as much as they can, and so on. Okay, so what evolution has done during multicellularity is discovered a way to bind these competent parts into a larger scale whole that and we can go. I have lots of sort of detailed hypotheses here, but the big picture is that the way that they're attached together enables the collective to do two things. One is that it enables it to have goals in a different space. So whereas individual cells have goals in metabolic spaces and things like that, large collectives of somatic cells have goals in anatomical morphous space. For example, they're going to make a correct limb, and if we keep cutting off the salamander's fingers, they will keep trying to get to that goal of having exactly the right number of fingers. So, so the first thing is being able to now work in a different problem space, and two is increasing the IQ of the collective so that you can have a bigger cognitive light cone. What I mean by cognitive light cone is the spatial and temporal extent of the biggest goal you can possibly pursue. Yeah. So if you're a bacterium, your cognitive light cone is very small because the only thing you can really care about is the local, let's say, sugar concentration, a few minutes of memory back, a few minutes of anticipation forward, and that's it. Your cone is quite small. If you're a dog, it's bigger, of course, but it doesn't extend 3 months forward. You can't get a dog to care about what's going to happen 3 months from now. You can't get a dog to care about what happens 10 miles away. Like the cone is.
That's why they're so happy, Michael.
That's right. Well, listen, you're onto something there because I think we are maybe the first creature whose cognitive light cone is bigger than our own lifespan. In other words, we have if you're a goldfish, and I don't actually know how long a goldfish's look ahead goes, but let's say it's a half an hour or something, right? Your goal of surviving that half an hour is pretty achievable. It's pretty achievable. For many humans have goals that are simply not achievable in their lifetime. That's it. They're just not achievable in the lifetime that we have. And we know this, and that may well give rise to some very challenging, psychological pressures that other creatures don't have because their cognitive light cones are smaller than their expected lifespan, right? That may be. I don't know. That may be. So the scale up does two things, right? It lets you work in another problem space. It expands your light cone. And so as these cells are working together on building or maintaining some complex organ, it has that has a failure mode. The failure mode is that whether due to chemical stress, drugs, mutation, whatever it's going to be, as soon as a cell partially disengages from that network, and it's electrical, but it's also chemical and biomechanical and some other stuff. As soon as it disengages from that network, it begins to be able to have a completely different calculus of rewards and risks. So at that point, while it's connected to these other cells, it cannot have thoughts of defecting because it's sharing through gap junctions. It's sharing its internal milieu, and your memories are my memories. And when we share memories, we don't have a real strong individuation. We're basically one creature. So you can't defect from yourself, really. But as soon as you start disconnecting, let's say by gap junction closure, and most strong carcinogens, and oncogenes, first thing they do is they close the gap junctions. And as soon as you've disconnected, your cognitive light cone, that barrier between yourself and the outside world, shrinks. Now the outside world, as far as you're concerned, the rest of the body, just the outside world, and so people often do game theory and they say, yeah, well, cancer cells are more selfish than other cells. No, everything's exactly equally selfish. Their cells are just smaller. They just have little tiny cells. That's all, right? Yeah. And that's led to our lab work on trying to expand that, right? To forcibly connect these cells to the rest of the tissue, and you can prevent cancer and do those kind of things. So I think that's the story.
In terms of the in terms of the gap junctions, it took me 30 years looking at your work before I actually realized, because I always got fascinated. I was counting them. I knew they were all there, but I didn't know what they were for. The physiology was telling me something else, but why do we have all these tight junctions that cap junctions all over the place? And then when I read your work and I said, oh, okay, this is really for, the enforcement of the circuit, right? You belong here. And this is the kind of, this is the kind of cell that you're going to be, like, you're going to be a skin cell, but when you take out that skin cell, you're going to become whatever organism you can envision yourself to be, which is like a Xenobot. I forgot to ask you in the Xenobot question, by the way, have you done it in other animals already, like taking off their skin cells or whatever cells and produce other alien life forms?
Yeah, I can. So yes, we have. I can't give details right now because it's not been published and peer reviewed. But the paper has been submitted. You will see it hopefully early this year, and it's kind of a dynamite story. So I think we can talk about it again once it's actually out. But the bottom line is that they look and act very similar to Xenobots, but they have some other capabilities that are pretty remarkable.
Oh, perfect. The other thing that I wanted to ask was, how far away clinically from using bioelectrical circuits in terms of preventing cancer cell metastasis and proliferation? You think how many years before we could actually use that technology in humans?
It's a good question. I'm not a clinician, and I don't have a super reliable way to predict what the FDA is going to do with these things, but I think it's a small number of years. I mean, we are actively pursuing that. I have a couple of startups that are beginning with the goal of approaching FDA with specific applications of these things. So I don't think it's 10 years, but I actually don't know how long these things really take. So. It's also, of course, it's a matter of funding and regulatory stuff, as much as the science itself.
Right. Tongue in cheek, I was saying, well, they should pour more research money into bioelectric circuits and start taking out money from too many DNA studies. But I will be pilloried for that statement, I know. But I've been complaining about that forever and ever because, for example, I encounter myself. For example, I am always deficient in vitamin B9, right? And it's because my enzymes are lazy and so on and so forth. So even if I found out genetically that I found out genetically that it's actually my genes that does that, I will still have to forever take vitamin B9, right? It's like, no, give me the solution, right? And that's what the genes are telling me. But is there any other way that I can do this without having to supply myself with vitamin B9 my entire life? And so that's where I'm coming from here. So, Michael, before I ask you the last question here, can you give us, you mentioned TAME earlier, and I loved your presentation on TAME. Can you give us a mini lecture on what TAME is all about and what you're trying to achieve there?
Yeah, let's see. The big picture is that I want to be able to recognize and relate to diverse intelligences. So I'm interested in all of the techniques, from starting with the conceptual shifts that are going to require us to define intelligence in a way that is not specifically tied to the very narrow examples that we've seen, so brainy animals. So when I am looking for a framework, I don't want something that works on humans and primates and maybe dolphins and maybe octopus, right? No, I want something that works on potentially alien beings, cells, tissues, software AIs, synthetic constructs, cyborgs, hybrids, you name it. We need to be able to be first of all, to know how to think about this. Second, we need a collection of tools to actually study and get answers within our framework of what to expect. And then we need a way to, and this is very important, we need a way to relate to them ethically. Because up until now, we've been using a very simple strategy, and we don't even use that well. But the basic strategy is something like this. If I show you a new system, what you can do is you can come up and knock on it, And if you hear a metallic clangy sound, then you conclude that, okay, this was made in a factory, it's not natural, which means it's not real, it's not really conscious. I can do whatever I want to it. I can take it apart if I want to, fine. On the other hand, if I do this and it's sort of soft and squishy and hopefully a little warm, then you say, I need to be nice to it, at least to some extent. If it's a cow, I still might eat it, but I'll go to jail if I do certain things with it. So this is like this criterion has been with us for the longest time, and it really it's based on making ethical decisions on two factors: what does it look like, and where did it come from? And I think we need to really wrap our heads around the fact that those are terrible ways to make these decisions. What you're made of and where you come from are not real. What you look like and where you come from are not great ways. That criterion is not going to survive the next decade because
does this contribute to our prejudice against the Turing test, for example? Is it contributing to our prejudice against the Turing test? I mean, I love the Turing test, but I know that people are prejudiced against the test.
Yeah. I mean, people are very worried about. Well, it's interesting. So people are very worried about being fooled and having false positives. They don't want to relate to something as if it had consciousness and then somehow find out that there's no one home and it was just faking, whatever that is. Now, of course, we know that from human history, we know that being too stringent about these things, in fact, inappropriately stringent about these things, leads to terrible ethical lapses because you can easily say, yeah, you're sort of human, but you're not enough like me for me to really care about what happens, right? We've seen this in human society from day one. So this, we're not even very good at being nice to things that do look like us. But if there are tiny differences, that's it. All bets are off. That's why people are they want to modify these functional tests like the Turing test with anatomical things. Well, when in a conference on synthetic biology, for example, or bioengineering, there will be an ethics seminar, and in the ethics seminar, it'll always be, well, does it look like a human brain or not? That's not cortex enough. We don't have to worry about it. Oh my God, this has the layers of the brain. We can't do it. Oh, the criteria is that it has to look like a human brain. I mean, this is ridiculous, I think, right? In the wide universe, I refuse to believe that in the wide universe, the only thing you have to ethically care about is something that looks exactly like today's modern human brain. I mean, it's just nonsense. So there's going to be I think, and the field of biorobotics and cyborg science and all this kind of stuff tells us that there's an enormous space of possible beings with radically different bodies and minds. They look nothing like us. That we need to learn to relate to. So that's the motivation for the TAME framework. So now, how do we do this? So what I was thinking is, what do all cognitive agents, and again, TAME at this moment doesn't say anything about consciousness. It talks about cognition, so publicly observable, just for the sake of getting some work done and not getting bogged down into philosophically unsolvable problems. For now, anyway, I focus on cognition, not on consciousness. So what do all cognitive systems have in common? I like William James's way of going about it. I think that all cognitive claims are really engineering protocol claims. So what do I mean by that? When you tell me that this system is at a certain cognitive level, what you're really telling me is a couple of things. What can I expect it to do when I'm not around? Is it going to do anything? Is it going to do the same thing? Is it going to do context-sensitive things? How cleverly, right? So the level of autonomy of this system, and what can I expect? What kind of problems can I expect it to solve? And the other thing that you're telling me is what tools in my toolkit can I use to control and predict this system? So, so in the TAME paper, I give just 4 waypoints on this continuum, but of course there are way more. And if I hand you a mechanical clock, then you are not going to train it. You're not going to convince it. All you can do is rewire it, really. If I hand you a thermostat and you think that all you can do is rewire it, you're missing out. But of course, if you think that you can convince it by rational arguments, you're also wasting time. So, right. So everything has an appropriate. So I call it the spectrum of persuadability. Now, of course, it kind of on the left side of the spectrum, it sort of all talks about control and persuadability. But on the right side of the spectrum, as you get to things like dogs, where you don't need to try to rewire their set point. You can train them with experiences, right? So that's better. And all
You're
the way
incredibly
on,
positive, though.
yeah,
You're being incredibly positive because you're saying spectrum of persuadability. I would say spectrum of stubbornness.
that's absolutely part of it. Resistance, right? So
Right.
you're right on, right? So resistance to different kinds of manipulations is a critical part of this. Knowing what can the system resist. So a type 1
Right.
thermostat doesn't resist you changing the set point. A type 2 thermostat has a metacognitive component on it that if you change the set point too much, it fights you on it. Cells do this when you try to change cell resting potential; they'll fight you on it. They try to resist, right? So anyway, over to the right of that system, where you get to rational systems that understand rational arguments, so humans, and then who knows what's beyond that. There, you're not so much just in the land of persuadability. You're really talking about relationships. So you could ask questions like, if I'm going to live the rest of my life on Mars, I'm going to Mars. What do I want? Who or what do I want to take with me? And what are the features of that system that are going to allow us to have the relationship that I want? Right? Do you want a Roomba? Right? You want a Roomba? You know. Maybe, but you'd like somebody that's a human level, and then they say, well, you know, I'm human, but you know, I had my couple of organs replaced with the electronic part, and you say, who cares? I don't care. And then you say, well, I had some, I've got some chips in my head. Meh, it's okay. You say, actually, I forgot to tell you, I'm entirely sort of synthetic. So the question is, what is it that you really care about, right? What do you really care about? And I think the key, I mean, do you care what they're actually made of? If, you know, if your spouse says, oh, by the way, I didn't tell you, but 10 years ago, I had all this replaced, you know, with other stuff. Is that a problem? I think what we actually care about is we care about the size of, and the size and direction of somebody's cognitive light cone. Because if they have the ability to care about the scale of things we care about, then we can have a particular kind of interaction that we couldn't have with somebody who has a much smaller light cone, and frankly, with somebody who has a much bigger one, right? So with a cosmic intelligence that is thinking about, you know, a huge number of things and can actively care and have compassion for millions of creatures in the linear range, the way we have with the 5 or 6 members of our family. I don't know if you could have the same kind of relationship, right, with a being who is so far ahead of you. So all of this is about understanding the degree of agency of the system, and it can take place, and it can have very diverse embodiments. Again, we're back to this. So what TAME tries to do is it first tries to shock by talking about all kinds of weird potential agents. It tries to shock us out of our complacency about only recognizing this sort of thing as something that you recognize having agency. Then it tries to offer some tools for how do we study agency in unfamiliar embodiments. And that includes building new ones, right? So that now that also includes a whole challenge of providing ethics. How do you relate to? And this is people sort of act as if this is a new problem, but science fiction has been on top of this for a couple of hundred years. If a spaceship lands on your front lawn and this thing on wheels sort of trundles out, you've got a real problem because right, and it sort of hands you a poem that it wrote along the way about how happy it is to meet you. And so what do you actually use to figure out whether you can take this thing apart or whether you're supposed to sort of be friends with it and be nice to it? So
military says take it apart, right? I'm kidding.
I mean, well, look, yeah, but I think a lot of people complain about. I've had a lot of people complain about Xenobots and things like that. These are the same people that eat burgers. So when you remind them that we take high level mammalian intelligences apart on a daily basis in ridiculous numbers and commercial food production, we are not good. Even with the puny kind of criteria that we have, we're not very good with it. So this is what TAME is all about. TAME is all about reframing our way of recognizing and relating to cognitive systems in our world, because I think for us, but certainly for our children's generation, they are going to be living in a world that if you watch the original Star Wars, that cantina scene at the beginning where there's every kind of weird alien and android, and this is, I mean, there's no way around it. This is what it's going to be. There's going to be every combination of evolved material, design material, and software is some kind of creature that someone's going to make at some point, and that's the world they're going to be living in. And we have to get over this idea that we don't know what to do with cognition unless it looks just like us.
Michael, you don't have to get into science fiction. This is just a point of levity. In order to for me, the way I visualize it when you were talking is that there was this beautiful woman in Virginia who got sued by her husband because the kids looked very homely, and it turns out that she had so much work done on her face and her body and never revealed it to her husband. And she actually lost the case, right? Because the case was about false advertising. So anyway, it's like, it's the ethics of the plastic surgery, et cetera, et cetera. We're looking at this also. That's why I remember, it's like, we don't have to look far, for what are the ethics of it. And, after that, the legal implications of it for us as humans who use the legal system for a lot of stuff that we fight about or that we don't like. But anyway, just hearing you say that in terms of the ethics of, all the things that we're doing, so you're going to Mars, what Xenobots are you going to take to terraform the system? What are, and it all begins to, my fascination here is that you have included the ethics in the entire process, right? Which is usually disregarded in many cases. For example, when we were doing lots of DNA manipulation, it was silly because we were doing, DEFCON 1 or whatever labs that had 4 different levels so that the organisms wouldn't escape and so on and so forth. And then now, like even just 5 years ago, we now have, DNA manipulation kits just being given out to high school students, and we don't have any DEFCON 1 stuff anymore. These are the kinds of things that I think about. That's why I think in terms of the TAME framework that you're doing, it's a lot in the biology and a lot of the thinking of the principles behind it, but also in a lot of the things that govern our core behaviors as a society, as a cooperative society. I think for me, when I look at the big picture, we need something like that in order to look at first principles, what really defines the whole cooperative system. And that's what I find fascinating with the framework. Although you proceed with very, very specific biological examples, I think the big picture thinking actually goes to the network of cooperative agents or competing agents that ultimately we have to exercise, a modicum of ethics in order to continue functioning. So, Michael, it's time for my last question, and it's a personal one. I heard that you homeschool your child. Do you still?
Now I homeschool the other one. The first one is now in college. He's a freshman in college. So we did, yeah.
That must have been a smarter, not harder decision. Now, as the last question, what 3 pieces of advice can you give our audience in order to live life smarter, not harder?
Oh boy, yeah. I
Take your
don't
time.
know. Yeah, I don't know that I'm competent to give anybody advice on how to live, but I guess I can tell you what I've done. One thing that is been really critical, and this goes, I started thinking about this when you mentioned the homeschooling. So the homeschooling was mainly orchestrated by my wife. Now, what she arranged, of course, was we weren't the only ones to the kid went to a million different classes and courses. He took college classes and various clubs and whatever. But she was the one who organized all of this. It takes a huge amount of organization, and I think it's a so I have a more general point to this, which is that what I consider myself to have been incredibly blessed to be able to do the kind of work that I do, and at the same time have kids and have a family and whatever. in no way would this have been all of those things together would have been possible for just me. Okay, impossible. So to me, the one thing I can, it's of course it's easier said than done, but I put a lot of effort also into arranging things this way, is to have a life partner that is aligned with you on how you see the various values and in practicalities of how you see that that's going to happen, right? So we were very aligned on the fact that we were going to have a specific division of labor, such that I was going to handle certain things, she was going to handle certain things. Without neither of us could have had the life we have. For sure, I couldn't have had this life without her. And so, to me, this is kind of very fundamental, because I see a lot of just observationally, I see a lot of misery from what by well-meaning people who are just not aligned in how they see things. And of course, I mean, I don't think there's any perfect solution. Of course, you can always diverge as you grow and all of that kind of stuff. But to me, one of the things that makes all of the various difficulties bearable is the fact that we are a holobiont of a certain kind, right? Together, we are tightly aligned on the important things. Lots of minor differences and on everything, but tightly aligned on the important things. So I guess you can blow that up bigger, which is to say that I think it's really helpful to surround yourself with a community that feeds the parts of you that you want to expand. So, and people often will say, well, in the olden days, it was great. You knew everybody in your village, but now with this electronic stuff, I actually think it's much better, because in the olden days, you would know some random people that you happen to have gotten, sort of dumped on Earth with, and that's it. Now, with the technological reach that we have, you actually can curate your environment. And right. Maybe, right, and you can actually say, what do I spend my time doing? Do I spend my time with people who make me miserable and angry on social media, or do I have a circle of people that I respect and that sort of feed my my intellectual aspirations, or whatever else it is. And so I think curating your environment is really is really key. And I guess the last thing I would say is really people call it knowing yourself and knowing what makes you happy, but I actually think it's less that and crafting and a little bit more crafting yourself, right? So the idea is, yes, you should know what your current skills and capabilities and the things that make you happy now, but I don't think you should assume that that's it and it's written in stone. I think you should have an aspirational version of yourself, and you should sort of work towards that and have a really clear, unflinching kind of look at what you've done, what you haven't done, and what you need to do in order to, get to sort of climb that Maslow's hierarchy as much as you can, right? Like, what's the top level goal here? Why are we sweating and bleeding? Like, what's it for? It's amazing. I get a lot of emails from just random people on the internet, and one of the biggest comments from people with really good careers and jobs and everything else, people are looking to have meaning in their life. That's maybe the most common thing that people say is that, yeah, I've got a job and whatever, but I want to do something meaningful. What do I do? And they're emailing me because they want advice on working towards some kind of scientific research or something. But that idea, thinking forward and saying, what is the meaning? And let's work towards that, as opposed to whatever, and that's easier said than done. But those are the things that come to my mind.
Sam Harris famously said that meaning is psychological question masquerading as a philosophical one, and I thought that was really, really funny, because you recalling that, because you said in terms of consciousness, right? What state are you supposed to be in? If what's the output of that? So I said, okay, the meaning. What's the output of that, right? So it becomes less of a philosophical question, and I think it's more of a psychological question. That's why you're in search of meaning, or for me, a coherent story, right? And the second thing that I actually love about what you said, into your second point, I remember there was a work of someone where most of the rewarding, many of the rewarding relationships that we have in life come not from our inner circle, but from the edge of our network. that provide us new opportunities and new ways of thinking, and that kind of openness to it's funny that this thinking came to me when I was working. I was programming an edge detection algorithm, for an MRI, and I said, well, let's see what's going on at the edge of this network. And when you look at that, I said, how does this apply to human relationships? So most of the beneficial surprise that we get are actually come from people that we hardly know, that are connected, by several degrees with us. So thank you very much for the three pieces of advice for living life smarter, not harder. And thank you so much, Michael, for being my guest today and enlightening us about. Really, this is not only groundbreaking work, but universe-breaking work, right? Because this becomes beyond space-time now in terms of the geometries that we're looking at. And it really requires a shift in the way we think now, and to look at different hierarchies of cognition and so on. And we're actually incredibly gifted with someone like you in terms of having someone in our species who actually says, well, okay, here's what we need to be curious about more. And in finding out really what are the principles here, as much as we can understand this universe. And with my mentor, Bill Yamamoto, he was the one who started the entire field of medical informatics. And he actually said, Ted, science is like this. It's like a wall that, beyond that wall, you don't know anything. And then you push it a little bit more, you know a little bit more, you push a little bit more. So it's not a process of, it's a process of discovering. So, and you are the one who, who inspire a lot of these younger students that I have that say, what else is out there that we can discover? So thank you so much for your work, and thank you so much for guesting our podcast.
Thank you so much.
I hope to talk to you again soon.
Absolutely. Anytime. Thank you. What a great discussion. Thank you so much.
Thank you.
This has been another episode of the Smarter Not Harder podcast. Smarter Not Harder is a major donor to Health Optimization Medicine and Practice Association, the educational nonprofit organization that teaches physicians and non-physician healthcare practitioners the standard of care in health, not the standard of care in disease. So can you help us by donating a bit of your time to spread the word? Head over to Apple Podcasts, Spotify, or wherever you listen to your shows and leave us a 5-star review. And while you're at it, please go to YouTube and click subscribe. Thank you for tuning in to Smarter Not Harder podcast, your home for one-cent solutions to $64,000 questions.